pleasure for me to introduce Rava da Silveira. Quite a few of us know Rava, among other reasons, because he's a regular instructor in a course in computational neuroscience in Woods Hole. For those of you who need the biographical details, Rava studied physics at the University of Geneva, and then he went on to do a physics PhD in MIT. And in fact, when I discussed with Rava, we realized that we are scientific cousins, because Rava's advisor at MIT, my advisor, did their own PhD with the same advisor. So his PhD was in statistical physics and soft condensation physics. And after completing the PhD, he went to Harvard University, where he was a junior fellow, and continued to work mainly on statistical physics, talking with people like Jean-Philippe Pouchot was there, and Daniel Fisher, and also people who, I guess, introduced him to neuroscience, Marcus Meister and others. And since 2004, he is in the École Normale Supérieure in Paris, and also, as far as I understand, from starting approximately now, a visiting professor in Princeton University, where he plans to spend a semester each year. And the topic of the talk, you can see it here. My drier translation will be that it is on the implications of correlations on coding. So.
Well, thank you so much, Cousin Yoram, for the introduction. As I've told already a number of you and of people I've met, this is not only the first time I'm at the Hebrew University, but also the first time I'm in Israel, and I have been enjoying every minute of it. So I think many of you will agree with me that sensory neuroscience has been focusing classically, traditionally, on correlating single neuron activity with either some percept or some task. And if you're trying to analyze the processing of simple percepts, then maybe this is enough. But when you go to more subtle or more complicated percepts, then a single neuron doesn't have enough coding power, doesn't have enough bandwidth, if you want. And one has to start thinking, as many of you here have, I gather, about how a population of neurons will process information. And when you think of a population, there are two aspects that come immediately to mind that are very different between a population and a single cell. And one thing is that different cells may be correlated among each other. And the other thing is that now that you have several cells, they can act in different ways. So the problem of how does a correlated assembly of cell process or encode information will be the problem I'd like to address. However, in my mind, it's a little bit of a technical corner, if you want, of a broader question, which is how to think about relations between perception and their neural correlates. And I could include a more general term or banner, if you want, on this left column, which includes also maybe motor tasks and not only sensory perceptions. And specifically, my focus will be that typically when you look at some neural correlate of some task or some perception, you will see it as being variable, whereas at least subjectively, we have the impression that we're doing things in a very certain way. When I look at you, I don't see a probabilistic listener, but I see a definite listener, a deterministic listener, or if you're listening. Now, so I don't have for you to introduce these. Excuse me?
You might be wrong.
Sure, but this is the feeling I have at least. So this is, I don't have to introduce these kinds of things for you, but just to kind of set the ideas. When I talk about variability, I'm thinking about these kinds of scenarios. So this is some experiment many of you have examined, and it's the response of the same neuron to 200, I think, 10 or 220 repeats of the same movie. And if you, of course, you see some structure, but you don't see a very dramatic structure, and in particular, if you focus on some little period of it and a set of cells, then you see very little structure.
Which area?
This, I think, is empty. So, was a test. So now, this is some kind of, if you want, for physics-minded people, in the physics vocabulary, you could say that it's some kind of annealed variability. But now you also have a quench variability, which is what I talked about as being physiological heterogeneity. So for Chaim, this is area retina. And you see the response of 150 cells, different cells over time. And you see that there are similarities among cells, but you see that there are also differences, very big differences among cells. So I'll try to address both aspects of this variability, the kind of dynamic variability and the quench variability in the later parts of the lecture. So the setup I want to have in mind is very similar to the Shadlin and Newsom experiment, but to simplify things, instead of thinking about a movie, let's think about being shown a very rapid succession of images, and on these images could be animals like a spider or something else. And every time I see an image, I'll imagine that some pattern of activity is elicited in the brain. Now, the first time I see a spider, maybe this pattern of activity is elicited. When I see the spider more times, similar patterns of activity will be elicited, maybe not exactly the same, and so on. So I can mentally associate this specific stimulus to a distribution of responses in some brain area, like retina or any other brain visual area. Now, if I show you not a spider, but a spider-like bug, then patterns will be mostly different, but some of the patterns may overlap. And so whichever area reads out from these neurons will have to kind of deal with this ambiguity. So clearly, if the situation is like this, so if distributions are kind of well separated in some space of neural response, then there's no ambiguity to deal with. But if we have the more typical situation where there is overlap, then one has to ask, one has to quantify the degree of this ambiguity. And this is what I'll do in this lecture, and try to say how correlations can affect how you quantify this. Okay? So, in some sense, what I want to say is that neural correlates or neural responses corresponds to pattern of activities that are variable, or at least if I look at any given neuron in the population, I'll see a very variable response. However, the percept is much more certain, and so there must be some reduced quantity that I can extract, some average quantity if you want, that is much less variable. So you'll know, for example, in experiments where there's the luxury of time, such as decision-making experiments, things like this, then an obvious dimension along which to average neural activity is indeed time. And so you get at some very robust estimations of what is going on in some populations as time averages. Now, in this scenario that I'm setting up for you, I'm explicitly ruling out the luxury of time, because I'm telling you you're seeing these things very fast. And this is, in some sense, to mimic real life. So if you're walking in the street and you see a crowd of people and trees and so on, then there's a very high, given your own motion, the motion in the world, there's very high stream of, a very rapid stream of input, and this kind of tries to mimic that. Okay, so if you're there, you don't have the luxury of time to average, so the obvious dimension where you can try to find reduced quantities is to average over the population. And what I'm going to try to argue is that one should really think of a set of neurons as behaving much more like an orchestra, where both the interplay between the players has some meaning, and also the differences between the violin and the trombones, contribute to the harmony. So basically, what I want to say is the meat of this lecture will be some proposal, or rather exploiting a proposal to try to argue that correlations can lead to very favorable aspects of coding. But before doing this, I want to make a few introductory comments, introductory steps, which will consist in just showing you very quick, a couple of slides on data, just to tell you what I mean by correlation, what are the quantities I have in mind. And then I'd like to argue that for this whole discussion to make sense, I have to think that I'm in a very low error regime. So I'm indeed seeing things with certainty, and I'll tell you what I mean by this. And just also I want to very quickly make some general arguments to illustrate the fact that it's maybe not so trivial a problem. Now, I gather from Chaim and others that some of you, I'm sure, are studying correlations. I gather you've done some journal club on correlations. So some of this thing may be too introductory. So if this is the case, just tell me and I'll speed up. Okay, so what I mean by correlation is always this. So the usual definition of correlation, some covariance between the activity of neuron I and neuron J, properly normalized. And from the data, I don't want to extract details, okay? I want to extract very coarse aspects of the distribution of correlation. So let's look at a few examples of data. This is in Chaim's favorite area, MT. And what you see here is an histogram of pairwise correlations. And what you see is that the mean is about 10%, okay? And it extends maybe to 30% in the positive direction. There is some negative correlation, some anti-correlation, but it's a small weight, okay? If you look at other areas such as V1, and you can do it kind of in some amount of detail by not looking at any pairs, but classifying your pairs as a function of their orientation preference, or what was it? Contrast preference, for example, then you can have more detailed information. But again, what I want to derive from this is not any kind of detailed information, but just the core structure. And repeatedly, the core structure is that the average value of the correlation will be around 20%. Most of the weight will be on the right-hand side of the axis, so most of the correlations are positive, okay? Now, there's not only in the cortex, there are other places where you have similar, we can derive similar conclusions. There is more recent data from a couple of years ago that present a kind of a different picture. And in that picture, the correlations that these authors find are much more centered around 0. They have a much more symmetric weight about 0. Now, these are measured differently. There are all kinds of controversy as to whether these guys are correct, the other guys are more correct. There are ways of doing the actual recording is different. The way of analyzing the recordings are different, so on. And I do not want to get into this controversy. What is useful for me here is that there still is some weight here around 10-20% that is non-negligible. Maybe a fourth of the distribution has its weight there. So what I want to extract from this data is that I will now forget about negative correlations. I will focus on positive correlations. I will say, let me think of a system of a collection of neurons that have positive correlation, and let me assume that the order of magnitude of the pairwise correlation is maybe 10%, maybe 5%, maybe 15%. I will not be interested in details of it beyond this. Any questions? Okay, so now one point that I really would like to emphasize is a conceptual point, which is that none of this discussion would be necessary if we did many, many mistakes, if we constantly did coding errors, or if I were thinking about an experiment where I've tuned the contrast to be so weak, or I've tuned the task to be so hard that the subject will be close to chance, for example. I'm thinking of a situation which maybe, or maybe not, but maybe is not so far from the real daily situation where you have very few hallucinations. You make very few perceptual errors. So let me try to quantify this statement a little bit. So imagine, I always say, imagine that you're lying down on your bed tonight and daydreaming about my talk. And if you're in Paris, this is the ceiling that you're watching. If you're in Paris, your room would be about 2 meter by 2 meter. And there could be a spider on the ceiling while you're daydreaming. And you would, of course, see that there is a spider, and you would see it very fast. Now, if you're in a Parisian room and the spider is maybe 2 centimeters, then there are 10,000 different spots where the spider can appear. Okay? And if, let's say, you take about 100 milliseconds, which is on the right order of magnitude, to recognize a spider, in 1 hour, there's about 36,000 events, or potential spider presence, if you want. Okay? So all in all, on this ceiling, during 1 hour, you could have around 1 billion spider events. Okay? Now, you're monitoring this whole ceiling with your visual circuits, and if you don't want to have hallucinations, so if you want the probability of error for each one of these events to be very low, let's say you want to have only 1 error total in an hour, okay, if you want this to be smaller than 1 error per hour, then you need, if in total you want less than 1 error per hour, then you need that each event is done correctly with a probability of error less than 1 over a billion.
But you assume there is no continuity in time.
Okay, so So at this point, I'm very upset that it's only Chaim who's criticizing me. At this point, you should all be telling me, no, there's continuity in time, you have priors, maybe you're in Scandinavia, there are no spiders, and so on. So all this is true. So you have continuity in time, the spider will not just materialize. Once you know that there is a spider in this area, maybe for the next 30 seconds, you don't have to monitor this area. And there are many other things. So, for example, there's maybe asymmetry between the misses and the false alarm. So maybe you want to reduce very much the false alarm, so you never want to hallucinate that you have a large spider on your ceiling, but you allow for you to miss it. So this you could change in many, many ways. Now you could, quantitatively, it could change in many ways. Now it could also change in the other direction, because you're not only monitoring for spiders, you're also monitoring for other kinds of bugs, you have other sensory inputs and so on. Now the point is, if you add all these things, do you change this by 10 to the 9 or not?
So suppose I'm daydreaming on the beach, and I'm looking at the sky, so
yes.
Then it will be 10 to the minus 20.
Fine.
You think this is?
Well, first of all, of course, I'm not saying that you're. So in the sky, you don't expect the appearance of a spider. You maybe would repeat the same thing with. So you maybe repeat the same thing with a bird and so on. Yeah, exactly. So you have to decide. You make it small. Would this diminish this by 10 to the 9 or not? My guess is no. So my guess is that this gives you. So, okay, let me just say, this is a kind of argument that says that if you do a very coarse argument, you get to these large number. If it were 10 to the minus 20, that would also be fine with me. And
do you think it's realistic to expect that the biological system, the real noisy brain, will generate such a position? This is realistic.
Why don't you ask me later on in the talk?
Okay.
I'm sure you will. So anyway, so basically here, what I want to argue, again, I don't want to make a detailed argument, and this number, I don't want to attach any specific value to this 9. It could be 10 to the minus 6, it could be 10 to the minus 12, and so on. But I don't think that to answer, or to give a preliminary, an interim answer to Chaim's question, I don't think that if you add these things of expectation and so on, and if you add every other detail in, like these various asymmetries, the priors, the costs, and so on, you will get this down to order 1. Okay. So what I have in mind, what the requirement I have in mind, to be very clear, is that I would like a set of neurons, a neural assembly, to code with a precision that is very, very high. And by high, I mean something like this, like 10 to the minus 6, 10 to the minus 9, 10 to the minus 12. Now, are correlations useful for that? So, before going to the more concrete part of the lecture, I would like to argue that it's very hard to make a back-of-the-envelope argument for it. because, so I'll make two back-of-the-envelope arguments, and they'll be contradictory. Okay? So the first argument is correlations are unfavorable. And you can all see this very easily. Suppose that I have some distribution of responses for the non-spider, a distribution of response for the spider, and let's suppose these were for independent neuron. Now let's say I include correlations. When I include correlations, the bigger the positive correlation, the flatter, the broader this distribution will become. And as a result, I'll make the ambiguity between the two much larger. Okay? So this seems to suggest that more correlation equals more ambiguity. And you can make this argument, maybe I don't want to bore you too much with this, so I'll go quickly, but you can make this argument slightly more quantitatively by saying, let's suppose that I have population with n neurons. For the sake of simplicity, I'll only allow them to spike 0 or 1 time, and they're all identical, so the state, if you want, of this system is given by the spike count. And with this spike count, I want to discriminate these two stimuli. So basically, what I want to see is how distant are the means of the two distributions as compared with their variance. So I can characterize, if you want, the performance of the code by calculating the signal-to-noise ratio-like quantity, which is the difference of the mean divided by the standard deviation, or its square. And you immediately see that if you don't have correlations, so you don't have this term in red, by adding neurons, you're doing better and better because the numerator grows like n squared and the denominator grows like n. Now, if you add correlations, obviously the denominator, the variance, will grow much faster, will grow like n squared, and at some point you hit a ceiling beyond which adding neurons is not favorable. Okay? So in other words, what this means is that if I have a correlated system, every time I add a neuron, instead of having the same increment in coding power or information, this increment becomes smaller and smaller. And at some point, by adding one more neuron, I don't gain anything. Okay? So this tells you correlations are unfavorable for encoding.
You assume the encoder, which is the sum of units, but there must be a difference.
Absolutely. So you're absolutely right, and this will be the resolution of this paradox. So I will get to it, but you're absolutely right. But here I've explicitly assumed identical neurons. But that's a very good point. I will discuss this in great detail. So now let me give you a very general argument that says that correlations are good, and there's various ways you can put it, but let me just say it in a very superficial way, in a very general way. So I said before that to each stimulus, I'll associate some distribution of the responses. Okay? Now, if I don't have correlations, what will characterize this distribution are just the mean firing rates. With correlations, I'll have additional numbers that characterize this distribution that are, for example, the values of the pairwise correlation or the endpoint correlation. So if I want to fit the stimulus with this function, so to say, I have many more parameters that I can use if I have correlations. Okay? So according to this argument, correlations should always be helpful. There's always some way of making correlations helpful. Okay? So these two arguments seem to contradict, and part of the resolution you mentioned, which is that neurons are different, they're heterogeneous, so assuming that just the spike count is the right variable is wrong. The other thing is here is how will I use these parameters to make my fit better, if you want. And this is kind of the proposal. So to fix the ideas, I'll be looking to quantify the figure of merit, if you want, of a code will be this ambiguity. Okay, so I will quantify everything I do as a function of this ambiguity, and most of you know that this is a good way to do it because this corresponds to what one calls the maximum likelihood bound. And if you calculate the error, the coding error, or as the sum of the misses, so these are all the patterns that you've interpreted as being non-spiders, but were in fact spider, and the false alarms, these are the patterns that you interpreted as being a spider, but in fact they weren't, then this gives you a bound on how on the performance of your decoder. Okay, so this will be optimal. So throughout, I will characterize the performance of a group of neuron, of a pool of neuron, with this number. So let's follow your suggestion, okay, and say maybe what I've done wrong is to assume that neurons are all the same. So what we did was to say, okay, let's go to the one step more complicated. What is the simplest model we can think about in which neurons are not the same? And this is a very simple model, which is you take a group of neurons, and a set of neuron, population of neuron, and you divide it into two pools, okay? And each pool, you'll assume that it's homogeneous. So the first pool will respond to the spider stimulus with some firing rate. The second pool with other firing rate, similarly to the non-spider stimulus. And mind you, here I'm not doing anything ad hoc. This is completely general because whenever I have two stimuli, I can divide my population in two halves that respond better or worse to one or the other stimulus. Okay, so there's nothing. So it's completely general. And on top of that, I'll assume that there is within-pool pairwise correlations and cross-pool pairwise correlations. Question? No. So now let's see what happens. And as soon as you calculate things, you see something kind of surprising happening. Okay? So this here I'm plotting this coding error that I just defined for you, this maximum likelihood error, as a function of the number of cells in the population. And what you see is that initially, when the number of cells is small, when I'm in a small population, the correlated value follows closely the independent value. That decreases exponentially here. That's a log plot. Okay? But fairly soon, when I'm around 40, 50, 60 neurons in the population, I have this very sharp plunge of the value of the error down to a very, very small, kind of astronomically small value of 10 to the minus 15, 10 to the minus 20. So the natural reaction when you see, and another way to see it is just to plot the ratio between the two quantities, and this ratio seems to be blowing up to very large values. So the natural reaction when you see huge numbers is to say, well, somehow this large number must have been put in by hand in the problem. So clearly I haven't put this large number in the population because I'm looking at a population of 50 or 100 neurons. So the population is extremely small on the scale of the brain or the cortex. Okay. So the other possibility is maybe I've put something very pathological in the values that I've chosen for my firing rates or for my correlation values. And you see this is not the case. The firing rates are 50%, 20%. The correlations are as small as 1 or 3%. So on purpose, we took values that are very close to the experimental values. So these are very harmless values. Okay. So what is going on? Well, how come I get such large numbers? So one way to do is you can do some analytical things and to try to understand it better. And if you do a very simple Gaussian approximation, you can calculate this error. And what you find is that indeed it will decrease exponentially fast with the number of neurons. And this is simply what you get for in the case of independent neurons. Okay, you can see this immediately. However, with correlated neurons, this acquires a denominator here. Okay, and this denominator will be some linear combination of the firing rates, the correlations, and the size of the population. And this linear combination will have this minus sign here, which allows it to become very, very small. Okay? So in some sense, if you want, in fact, as you increase the number of neurons, this looks like essential singularity. So in other words, this will suffer from an essential singularity. So in other words, the scaling here is not what is changing. What is changing really is kind of the prefactor, if you want, in the coding. So this is the mechanism that is going on. Now, yes.
So it diverges at the finite end?
It does, if you fix the correlation values. But
this means that there is some constraint on the
yes, yes, absolutely. So okay, so I can give you two answers to this. One is a mathematical answer, and one is kind of a more conceptual answer. I think let me start with the latter. Conceptually, there are two different problems. One problem conceptually is if you have a large population, and now I look at a small part of this population, and I ask what is the value of the pairwise correlation, what should the interactions in the large population be to get these pairwise correlation values? This problem I have not solved. I take these values as given, okay? Now, it may be that in the brain these interactions are such that the correlation values that you get are in fact extreme, even if numerically they don't look extreme, okay? Now, so I take these as a given, these correlations, and I fix them. And then I ask, you know, what is going on? Now, mathematically, what does this divergence correspond to? It corresponds to when some patterns have a probability that goes down to 0.
Yeah, but
so you forbid states, basically.
If I use n larger, then it's meaningless.
Yes.
What?
Yes.
It is meaningless.
Yes.
But how come? The problem is not defined.
What do you mean?
How come you get negative epsilon?
No. So this means that that you couldn't have a population with this number that has that for every neuron. Absolutely.
Yeah, so
absolutely. But it doesn't. No. But first of all, the important. So okay. First of all, the important thing is not that it diverges. The important thing is that you get to small values. So it's not. I'm not kind of attached to the fact that it diverges and that some things go to 0. But the question is, indeed, if in reality you have a piece of cortex that will have a distribution of these pairwise correlations, what this means is that if something like this is true, then you cannot find in this population a subgroup that will have all the in-pool values close to C1 or C2 with an n that is larger than n prescribed by this, okay? But it doesn't mean that you can't get close to it. Doesn't mean that there's not a subpopulation that will where something like this will behave. And okay, so maybe I didn't plan to discuss this, but you can actually make some heuristic calculations and say if I take the data for, let's say, V1, now I can estimate within, say, if I look at 100,000 cells or 1 million cells, within those million cells, how many subgroups will there be where something like this can happen? And you can estimate these numbers from the data. So if you want, I can describe this a bit more, but I wasn't planning to. Okay, so more pictorially, what goes on, I think you can explain in this way. So again, what I had assumed is that I have a population with 2 subpools. Each pool is homogeneous. So the first pool is described by some spike count, K1, and the second pool by another spike count, K2. And so the system as a whole is defined completely by a point on this plane, K1, K2. Okay? So if you have independent neurons, in response to a stimulus, they will give you some distribution. So the mean, let's say in response to spider, the mean response will be here, but then you have fluctuations around it from trial to trial. And these fluctuations, if the neurons are independent, will scale as the standard deviation will scale as square root of k. So you have these kind of roundish clouds around the mean. And with these roundish clouds and kind of finite number of neurons, you have no other solution than to find these overlaps. Okay? But now if I correlate neurons across the pool, then these objects will kind of elongate. And they elongate in such a way as to decrease the overlap. So they elongate, if you want, in the right direction. Okay? So a couple points here. So first of all, the correlations will not shrink your distributions. They still elongate them. So if you look, for example, at the projection on this K1 axis, the overlap of the projection here will be bigger than there. Okay? So if you just looked at the activity of a few neurons, say in K1, and you tried to decode things from there, then you wouldn't get, you'd get a worse outcome. And conceptually, what is happening here, the mechanism, if you want, is that the correlations, are shoving the noise in this direction. So they're shoving the noise in a dimension that is irrelevant to coding. And they're taking it away, if you want, from the dimension that is relevant to coding. Now, this mechanism is not new. It existed in the literature. It actually existed before the papers that we often think of as being the first papers. Already in the early 80s or late 70s, some people proposed such kind of mechanisms. And this idea of having correlations shove the noise in irrelevant dimension has many incarnations. Chaim has some very nice results in the context of ring models, where the same mechanism can be exploited to understand a number of things. So the mechanism itself is nothing new. But I think what the new thing here is to notice that when you implement it in this way, with this kind of structure of dividing the population in subpopulation, then numerically you can get to a very, very large effect. Okay, yes?
If the means were organized in some other way, the noise would
yes,
be mixed force.
that's right. So let me... Right. Okay, so okay. Yeah, so I wasn't... Again, this is some of the things I was not really planning to discuss, but since you ask... So basically this is the picture. So if you want negative signal correlation, you want positive noise correlation, and vice versa, okay? The reason why I'm looking at this situation is that, first of all, I'm assuming positive correlations in pairwise correlations everywhere, and second of all, I have a situation where I've divided my population into subpools that are more or less responsive to spider. So this, for my example, was the natural situation, and I want to focus. But conceptually, they're all the same. I mean, it's just a matter of how you structure your matrices. Okay. So, yes.
Can you just I don't get why when you put in correlation the circles become ellipses.
Okay, so basically here, one way to think about it, if you want, imagine you have some Gaussian distribution, okay? So the correlation will define you this quadratic form, okay? And depending on the value of the three coefficients, you can deform this correlation in various ways. So what I'm saying here is, if maybe I should have described this a little bit more in detail, but what I'm saying this is that for some patterns of correlation, and specifically patterns where C12 is a little bit larger than C11 or some combination of C11 and C22, then the distributions will deform in this way. Okay? Indeed, they could deform in a different way, but there are classes of correlation values for which they deform in this way, and those are the classes that are useful for me. Okay? So now let me just exploit this a little bit farther, kind of to see numerically what are kind of the limits of this. So let's push the problem a little bit more, and let's really want a small population. Does this still work? Does this mechanism still work? So here we're looking at really tiny populations, or a few neurons, or 20 neurons at most. And what you see is that even for such small population, for firing rates that are kind of acceptable, you don't have to push the correlation values beyond 20-30% to get to these tiny, tiny values in the air. Okay, so that's just a numerical observation, kind of a quantitative observation. And the most surprising, along these lines, to me, the most surprising is what happens if independent coding completely fails. So now I'm thinking of a situation in which the mean response to the two stimuli is tiny and differs, called delta, this difference differs by 1 or 2 spikes, okay? So if that's the case, if the distance between these 2 points is 1 or 2 spikes, as soon as I have a population that is somewhat appreciable, the information will completely be drowned by the fluctuations. But even in this case, correlations that are fairly small kind of are enough to decrease the error, the productive error for populations that are kind of still very small on the scale of the brain. Okay, so this is just to illustrate that numerically is kind of a powerful phenomenon. Now, maybe I will skip this because so you could ask a question of robustness. Maybe some of you have this in mind. So when I did this, you could ask, oh, well, you know, what if they're not exactly parallel? What if there's some differences in the correlations and so on? And you can study that, and the upshot is that things are fairly robust. But let me skip this maybe to get to more conceptual things. So until now, what did we do? Until now, we said, okay, let me fix the stimuli, the set of stimuli. I have two stimuli, and let me minimize the coding error, okay? So now I want to turn the problem completely inside out and pose it in this way. So let me think now that I'm fixing the error, and I'm asking, given some error threshold, how many stimuli can I encode such that I can discriminate between any two stimuli with less than this error, okay? So really what I'm asking here is not a question of discrimination, but a question of capacity. And for the sake of precision, let me define capacity as the log of the number of stimuli that you can code faithfully divided by the number of neurons. So this is the capacity per neuron, and I define it like this so that if you had deterministic neuron, then you would have 1 bit per neuron. So that's the natural definition of capacity. Okay? So let's go back to our example population with 2 pools and ask this question. Okay? So what does it mean? What it means is I want to fit many stimuli, so I want to fit as many distribution of activity as possible within this plane of spike count K1 and K2. However, I want to fit them in such a way that they don't overlap, they won't overlap much. I want the centers of the distributions to be separated enough such that I have a I can have faithful discrimination. And the fact that these are kind of roundish will prevent me from fitting many of them within that square. By contrast, the correlated distributions can become very, very thin, so I can stack them with their centers very near from one another. Okay, so I can stack, if you want, many more. So it's kind of interesting. You can estimate how many of these you can fit, and in the independent case, the number of distributions you can fit will grow like n. And the reason it grows like n and not n squared is that, as I said before, this standard deviation goes like square root of k. So you lose a square root of k if you want along this axis. You lose a square root of n along that axis, and you end up not with n squared but with n.
Well, n is the number of neurons.
Yeah. So here it's 200 because in each pool I have at most 100. Yeah. Now here but in addition to this, you want the tails not to overlap too much, and this will give you this prefactor. And what's kind of interesting here is that if epsilon, you require epsilon to be very, very small, like 10 to the minus 6, 10 to the minus 10, then epsilon square will be very, very small and will dominate completely over the n terms. In other words, if I look at this capacity for different size of the population, when my error becomes very small, becomes of the order of 10 to the minus 6, 8, 10, then whether I have 10 cells or 100,000 cells won't make any difference, essentially, for the capacity. So what dominates here really is the error and not the size of the population. Okay? Now, there's no reason, there was a natural reason to divide my population in 2 pools when I was faced with 2 stimuli to discriminate. But now, if I want to discriminate among many stimuli, there's no reason I should stop at 2 pools. I could think, for example, of 3 pools, and if I have 3 pools and I have 3 numbers that define my system, the spike count, K1, K2, K3 in each pool, and now the question becomes how to fit distributions not on a plane but in a cube. And as you'll know, it's much easier to fit cucumbers in a box than to fit melons, and as a result, I'll have a much higher capacity again in the correlated case, and I can generalize this to a general dimension. Okay, I divide now my population into D pools, and what I find, and one can calculate everything, but okay, let me not spend too much time on that. So what you find is the more heterogeneous, so the more divided your population, the better it becomes. Here I'm plotting the ratio of the capacities. Actually, I think here is the ratio of the number of states you can code as a function of the neurons. So the bigger the system, obviously, the better, but also the more heterogeneous, the better. Yes. Okay. So that let me get to the next slide, and I'll discuss this briefly. Okay. So now here there's something that I'm not doing in a very clear way, which is that there's no reason I should compare the case D equals 3 for correlated and independent. Okay, what the more natural or the more defendable comparison is rather comparing the optimal D, the result for the optimal D in the correlated case and the optimal D in the independent case. So this is what I do here, and that again is kind of interesting. Is that so? Let me, instead of describing the result as a function of the dimension or the number of pool, let me describe, because it's more natural, the result in terms of the size of the pool. So this small n is the number of neurons in each subpopulation, in each pool. Okay. So if I have an independent population, then optimally the pools have to grow like the error threshold and have to grow logarithmically with the error threshold. Okay, and as a result of this, the capacity will be suppressed like the log of the error threshold. Okay, this is because you want some averaging in each pool before you put the pools together, in some sense, intuitively. Okay. By contrast, in the correlated case, the scaling will be as follows. So to answer your question, I have to go a little bit beyond the scaling. So I will mention this and then I'll come back to your question. So the scaling in the correlated case is that n is fixed and will not anymore depend on the error. Okay? And as a result, the capacity will also be fixed. So the capacity per neuron will be fixed. So what it means really is that I'll use up some number of neurons, and this will give me one of these elongated objects. So it's like kind of a mega neuron, and this mega neuron is deterministic. Okay? So here the scaling is a scaling that suffers from the fluctuation. Here the scaling is the same scaling as in the deterministic case, only instead of having capacity per neuron being 1, the capacity per neuron is a fraction of 1, because I'm using many neurons to make kind of one mega neuron. Okay? So now how I choose the correlation, I retrieve to this image, if you want, here. And what I'll say is, okay, I fix some error threshold. Okay, this is the first step in the construction. Once I fit some error threshold, then let's say I allow some distance between these, between the means of the distribution. Okay. Given this distance, I have some range of correlations that will be acceptable. That will give me error less than this threshold. Okay. This is the range of the correlation. Okay. And now, depending on how I choose this, I'll change, if you want, the numerical value of this quantity. So if I were to do a calculation that were kind of more correct or more detailed, I could express this as some kind of very weak dependence on the error. But for all practical purposes, I can think of this as being fixed. And when I say between 0,1 and 0,3, this is because I have in mind cases where the distance between the means can be as small as a couple spikes or a few spikes. And yeah.
Do you have an actual procedure for partitioning the sort of neurons to the groups?
No. Okay. So here we're just thinking of something completely simple and idealized. Yeah. Sorry. Okay. I'll come to this also. Maybe there are some results I'll show that will satisfy you a little bit. Okay. I will generalize this to include more realistic kind of division of the population, if you want. Then come back to it if you're not satisfied. Any other questions? Yeah.
You skipped the decoding phase. The likelihood of the stimuli. What is the decoding procedure?
So here,
the decoding from the.
So it's the same. So I'm saying given. So if you want, the figure of merit is always the same as this optimal decoder, this maximum likelihood decoder. Okay. Okay, so let me come to your question about is that realistic? So again, what we did here was really to cut the population kind of in this idealized way. Each pool is homogeneous, all pools are equivalent, and so on. And this clearly is not correct. So if I look at the population of neuron, not only maybe I can distinguish pools, but really I'll have neuron-to-neuron variability that are appreciable. Okay, so so I think one can just write down the problem much more generally and just look at the distribution of firing rate and distribution of correlation values, and just write down the decoder and find very similar quantitative results, but the structure of the distributions will be very different. So ideally one would want to do this. We haven't done it, but I think it's possible to do, and I think that would satisfy you. Now, what we have done is to say, well, let me generalize a little bit this problem, and instead of thinking that each pool has a given firing rate, which is homogeneous, and given, say, in-pool correlations which are homogeneous, let me perturb these. And let me assume that within the pool there's a distribution of firing rates, there's a distribution of correlation values. Okay? So I want to say, now if I include neuron-to-neuron variability, does it destroy my construct? Does it fail, if you want? And what we found is that in every instance that we examined, it makes things better. Okay, so this is a summary. This is the degree by which you perturb, if you want, your parameters in your model, and this is the error suppression. So this is by what factor your error becomes yet smaller. Okay, so the more you perturb your values, the more the error gets further suppressed. Another way to represent the results is that here's a histogram where for a given perturbation of 2%, we tried a number of instantiation of the population, and in all cases, the suppression was to the right of 1. So in all cases, you get a smaller error. And if you repeat this with a larger distribution, then you find that this histogram moves to the right, and maybe the weight is around a factor of 5 or 10.
So isn't heterogeneity like the number of pools in N?
So again, I'm not sure what is your question, because
I'm saying if
it should have been answered already.
Neurons are different.
Yes.
So that's basically said that I have N pools.
No. Because you want, so if you do this, you will not capitalize on this pooling effect, on this averaging effect. You need some form of... Yeah. So I said, so that's right. So, yeah.
Will do whatever it needs.
Absolutely. So that's what I said before. I think in answering your question, what I said before is that in general, you could write down the problem like that, and the only question there is to find the decoder, and I'm sure you can do it. Yeah. So I'm sure you can do it. Yeah, definitely. I'm sure you can do it. You can write it and so on, and you'll find numerically similar results. But now we just did this as a kind of El Cheapo version of that, if you want, or a proxy for that, which is just to perturb the pools. But no, I agree with you. Okay. So now, just before I conclude, let me just depart a little bit from my discussion of correlations. And since we notice this effect here, which is that if you take a system which has some degree of heterogeneity, you break it up in N pools, and then you perturb it to get neuron-to-neuron variability, your results become better. One can ask the question, how general is this result? How is it true that heterogeneity will always help you? And so we examine this problem in various ways, and the answer is always yes. So let me just show you a couple of ways you can approach this, and then let me conclude. So here is a case where we started from data. So we looked at a retinal recording of 111 cells, and we compared different systems. So we compared the actual system coming from data, with the numbers that we could derive from data, and then we compared it with systems that mimic the data, but instead of being completely heterogeneous, we transform them into 4-pool models, 2-pool models, and 1-pool model. Okay? And what we find, so if you want, this is kind of the reverse of the exercise we did before. And the only advantage here is we're using actual data, so maximum likelihood decoding on actual data to give us some kind of limiting behavior, if you want. And what you find is that both the slope and the let's say it becomes systematically better. The error goes down systematically as you go from a less heterogeneous system to a more heterogeneous system. Okay. Yes. So this is the case d equals n if you want. Excuse me?
Which plot does it derive?
This one.
But you can see that the other colors are from the simulation, or?
Yes, so the other colors is we take, now what we do here, we divide this group of cells into a number of pools, keeping some constraints. So the constraint we keep is that the overall firing rate should be fixed, because we want to compare things that have, say, the same metabolic cost or something like this. So we keep one quantity fixed, and then we approximate the system where, if you want, d is equal to n, in a system where you have 4 pools. Okay? And then we compare their performance. Okay? Now, you can also, now forgetting about data, you can actually do derive purely formal results in various limits that show that heterogeneity is variable. So let me just quickly mention this, but let me not dwell too much on this. So one thing that you can do is to look at the completely general case where you take a system with uniform firing rates and you perturb them. And this index should be there. And what you find is that systematically the error goes down. Always the error goes down. What you can do also using convexity properties, you can show that if you're looking for rare targets, like if you're looking for a spider that is very rare among other stimuli, then the error in a heterogeneous system is always smaller than in a homogeneous system. And this is just completely general, just using convexity properties. So in any way we kind of approach the problem, it appears that heterogeneity is helpful. Yes?
But into stimuli, I would suppose that if we have 2 pools, then the system is linear, so the error should be smaller. But my intuition is wrong.
So I'm not sure I have a very good kind of intuitive answer. Let me give you 2 possible answers. I'm not sure they will satisfy you, but this is it. So, as I said, something you have to keep fixed to compare more or less heterogeneous systems. So let's say you keep fixed the average firing rate. So in one case you have every neuron that fire at, say, 70%. Okay? And then you divide them in 2 groups or in n groups, but let's say you go from less heterogeneous to more heterogeneous. And then you'll have neurons that now you divide them, so now half of the neurons will fire 60%, half of the neurons will fire 80%. Okay. So the ones that fire 60% now will carry less information. And the ones that fire 80% will carry more information. But now if you look at how the decoder works, the ones that are 80% will be more effective. So you gain more by that subgroup that has gone to 80% than you've lost by the group that has 60%. Okay? So this is one way to intuit it. Another way, which is kind of more superficial, maybe more literary, is that if I'm trying to code for an ensemble of inputs, if I have only identical sensors, I will miss a lot of the information. But now if I have many different sensors, then I can kind of capture non-overlapping sensors if you want. I can sample the information better. But that's not a very solid way to express it. Yes?
The final rate is limited between 0 and 1, right? But your heterogeneity is the Gaussian, right?
Yeah, so we took... That's right. So I can't remember if we clip it. I think we just take small enough value so that it doesn't spill too much. Or we clip it. I can't remember whether... I think we just...
One side, then increasing the heterogeneity can increase the mean.
Yeah, that's right. So I think it's small enough so that we don't have to worry about these things.
But in the plane where you show K1 versus K2, can you explain the intuition why heterogeneity works? Because it's just changing the place of the stimuli in the plane, you know?
Yeah, so that's right. So let me see. Again, it's okay. I don't know if I have. So the way you have to think about it is that now. Okay, so let me go back to this drawing. I was thinking of how to draw it. I don't have a nice way to draw it. I should think more to find a nice way to draw it. But basically, now you're dividing this into kind of subgroups, if you want, and this one too. Okay, and so now you have to draw it in higher dimension.
Let's say that just the fine rate of pool 1 is different than the fine rate of pool 2.
No, the fine rate of pool 1 is different from the fine rate of pool 2 in this case. So now what I'm saying is that I'm perturbing the mean of every neuron. And again, I think the way to have the intuition is to what I was saying before. So now let's suppose that now pool 1 you divide into two pools, and half of the neurons are spiking a little bit less, half of the neurons spiking a little bit more. And if you find the right way to draw it, you see that the ones that have more extreme value will kind of serve you much more for the decoding. Okay, I have to think to find a better representation for this specific result. Okay, so let me conclude. Okay, so let me conclude by first summarizing a few points and then mentioning some open questions. So I think the first thing, which is completely independent of any results, is I think this regime may not be completely uninteresting. People like me that come from physics have this disease, this kind of Tourette syndrome of always wanting a large number and perturbing things around this large number, and the obvious large number in the brain is n. And I'm not sure at all that this is a legitimate way of thinking, and maybe what one should think about is very small circuits, and however, maybe there is a large number, and this is kind of the performance of these small circuits. Now, so the main results that I mentioned, that I discussed, was that you can get very strong suppression of the decoding error and similarly massive enhancements of the capacity by this simple effect that was noticed before, but if you kind of capitalize on small population, on pooling on small population. And basically, why does this numerical effect doesn't happen in many models that were studied before, is that when you don't capitalize on pooling effects, when you have, say, neurons that are very different from one another that make the population, then to get a large effect, you really need the correlations to be of order 1. But when you have a pool of neurons that are somewhat similar in their properties, they act like one big neuron, and what you need really is that just not the correlation be of order 1, but n times the correlation be of order 1. And so if if the correlation is 10%, n just needs to be of order 10. Okay. And the last things that I discussed was some results pertaining to the fact that whenever I have greater diversity, greater heterogeneity in my unit, if you want, the coding performance, the effect becomes stronger. And finally, I skipped this. I didn't discuss why the results are robust. But there is something that one of you asked about, which was, why I have to choose these correlations in such a way that they will elongate that will deform, that will distort the distribution in the way I want. And indeed, I need a special class of correlations. And one can have a fairly detailed discussion of whether these correlations are natural enough. And if you look at, say, data in V1, your knee-jerk reaction would be to say that they're not natural. That's not the kind of correlations you would expect to find if you looked at, say, V1. And then one, as I was mentioning maybe in answer to Chaim, one could ask questions slightly more quantitatively, and let's say if I have a population that looks like V1, how likely is it that I'll find in a subgroup of, say, 20 neurons some structure like that? And one can do this a little bit more quantitatively. Okay, so this is just, as you've seen, kind of very idealized questions, very idealized models. There are many more things one can think about. There's questions of stimulus dependence of the correlations that some of you have worked on. As some of you have pointed out, maybe you want to have a much more natural definition of the model that doesn't have pools or things like this, but includes a more functional form of diversity. There are many mechanistic questions, you know, are these correlations that are all the time in the network when I measure, or do they suddenly emerge after some transient stimulation of a network, for example, things like this. There are lots of questions about reading out, which in my mind are the more difficult questions and things I haven't thought about yet. Maybe the most exciting are experimental questions. And the first thing I would do, and maybe we're thinking of doing this with data, is to take some data where you have an animal that does some kind of discrimination task, or you have an animal subjected to two different conditions, and you look at the simultaneous activity in a neuron assembly, and then you ask, you know, what kind of classes of correlations do I see? And this is, again, not a completely trivial problem. If it were only because you may be looking for a correlation in groups of 20 neurons, and you may have simultaneous recordings of 100 neurons, and so just to search for these groups is itself not an obvious problem. Okay, so there are many further discussions to be had. So let me conclude here, and before ending, thank my collaborator. All the aspects of this work were done in collaboration with Michael Berry, who's at Princeton University, and a number of things also were done together with students at École Normale and at Princeton University. Thank you very much.
Time for a few questions. Any questions? Yeah.
A few questions. So you're pulling basically a linear data with 0, 1 values. Is it because of 0, 1 values?
No, okay, I should have mentioned this. That's a very good question. So I should have mentioned this. The 0, 1 is just for the sake of simplicity, just to simplify my life. If you have, okay, so if you take more than 0, 1, let's say you allow them to spike, much more generally, if you do a kind of approximate treatment of it, it will just make the effect stronger. If you want to do a more systematic treatment of it, it becomes more difficult, because now each pool is not described by one number, but is described by some vector, if you want, of how many neurons you have that fire how many spikes. So the treatment becomes a bit more difficult. But conceptually, it doesn't change anything. Indeed, conceptually, it makes, numerically, it makes the effect stronger.
So I'm still confused about how you model in general the population statistics in the correlated case. Is it Gaussian correlation?
Okay, so that's a good question. So we did, okay. So what is the given of the problem, and then how do we treat it? The given of the problem is we just take these pairwise correlation values.
But you need P, you need probability.
That's right. That's right. You're absolutely right. So what we write down as assumptions are these values. Then we need P to calculate the error. So this we do in two ways, and we did in two ways to kind of cross-check them. Now, how would you want to do it ideally? What you would want to do ideally is to calculate the minimum mutual information, okay, and then to do the decoding with that distribution, okay? Now, we didn't do this, and so we did two things. We did maximum entropy and Gaussian, okay? But let me, because one thing that was kind of so the maximum entropy was because we thought, it's broad and so on, But one thing that was surprising to me is that you were very much in the tails. So I expected that maximum entropy and Gaussian would give you very different results, but they match.
Yes, you said that this coding trick works when the sign of the signal correlation and noise correlation are opposite. Right?
That's right.
Whether you have negative and positive or the other way around.
That's right.
What are the experiments say about that? If you measure two neurons, is it more likely for positively signal correlation neurons to be negatively noise correlated?
Okay. So I don't know I cannot give you a catalog of answers. I don't know a catalog of answers in different brain areas, and I'm not even sure this has been systematically studied in different brain areas. One place where people have studied this somewhat systematically is V1. And in V1, if you ask your biologist from the streets, he will tell you that it goes against this. Okay, so he will tell you that neurons that are that have similar preferred orientation are also more correlated. Okay, now this effect is weak, and recently I was talking to some experimentalists in. I don't know if the paper is yet submitted, so I'm not sure I should. It's published, so I'm not sure I should describe in detail, discuss this in detail. But according to recent experiments, there are cases where actually the reverse is true in V1. Okay, so it may be that by looking more in detail at the data, you would find that it's not as clear-cut, and you would have both cases depending on which neuron you look at and so on. But regardless of all this, I think in my mind the question is much more a question of readout. Because if, first of all, I don't think that exactly this will happen in the brain. I would like it, but I think the chances are extremely, extremely low. But if something kind of like that is going on in the brain, one result that these studies suggest is that you don't need to look at 1 million neurons or 10,000 neurons, maybe 100 neurons. So if you're down at 100 neurons, then it's not so important what is the statistic average over all V1. What is important is how to choose those 100 neurons you're going to read out from. Okay? And then the question is, how many groups of 100 neurons within V1 are there that would satisfy some correlation structure like this, even if you have, on average, the other one. Okay. And that you can calculate, so I can show you.